James had a major contribution to cellular trafficking, and in particular how vesicle, which is really a quantum of cellular compartment that can contain many proteins, hormone, neurotransmitter, is transported from one part of the cell to another part. So James was awarded the Nobel Prize in 2013 in medicine and physiology. So this transport underlies many cellular function, and although many molecular information are already known, how a vesicle goes from one compartment to another remains unclear. And so we thought, when organizing this program, that really the field of cellular biology became mature enough for the mathematical sciences and the physical sciences, so that modeling would be something important. And so we have Jim here as a part of our effort, really to present this new question and problem about cellular biology to the mathematical and physics community. So James, we're very happy to have you here. Please.
Yes, I have to get wired up. Good. Okay, well, can you hear me okay? Yeah. It's on. Okay, good. Well, David, thank you very much. I met David in Paris, where he's known as David. And after an evening lecture I gave at École Normale Supérieure for the public, and there were almost all lay people there, and I had expected very few questions of the specificity that I got from David, who came up to me with an incredible passion as it regards the ion calcium. And I've learned more about calcium today, even in the last hour, than I may have in my lifetime. But I was very happy to learn it, and I had a great time at lunch, and I'm looking forward to talking to more of you afterwards. I think what you are doing here is very important because so much of probably many fields, but certainly cellular biology is becoming understood and is understood at increasingly at a biochemical and a biophysical level, and certainly the problem that I'm working on, and that I'll focus on today, well, one of the major problems I'm working on, concerning synaptic transmission is an example of that. And we've started to collaborate with people that, like yourselves, or many of you who are engaged in modeling activities, and I hope that we'll be able to do more. I must say the building here at the Newton Institute is very impressive. It's designed for interactions. I did have kind of a question about the kinds of interactions you expected in the men's room. I noticed there was a blackboard there, and I thought that meant that people have meetings there, but I really don't know if I should speculate about that any further. Okay, well, so knowing that people have a variety of backgrounds, not all in biology, I thought I'd provide a little bit of a general introduction to synaptic transmission. I think everybody here knows that nerves conduct electricity and through action potentials, membrane potentials, and when the action potential, the neuronal impulse reaches the end of the wire, so to speak, it needs to be passed to the next nerve in order for a neural network to be created that functions and which engages in computation. And I think you also know that each nerve is impinged on typically by many nerves, some of which have a stimulative excitatory effect, others an inhibitory effect. But the contact points in every case have the same molecular anatomy, and they're called synapses. And on the presynaptic side, there is where the end of the wire, so to speak. There are vesicles that are found at the very end in what is called the active zone, and those vesicles, as Bernard Katz discovered many years ago in London, contain the neurotransmitters. And so Neurotransmitters are, of course, the chemicals that are introduced into the gap between the two nerve cells, the synaptic cleft, and which diffuse across the synaptic cleft to activate receptors on the other side in the target cell, which then reinitiate or lead to a process of reinitiation based on the computation of the of that cell. Now, a very important aspect that is sort of underlies the thrust of the very specific problem we're working on is speed. We know a lot about the mechanics and the biochemistry of how a vesicle fuses with a target membrane, whether it be a synaptic vesicle at a nerve ending or any other vesicle in cell biology. We understand a lot about that. But those fusion processes, in general, they take, 100 milliseconds, a second, somewhere in that range. And that's really fast for most things that have to occur in the body. For example, the release of insulin into the blood is one of the fastest responsive processes in the body. If you eat a meal, your pancreas very quickly, after receiving a signal, appropriate signal, will release by membrane fusion stored insulin into the blood. That process begins within 100 milliseconds, and it's more than adequate. Almost every other fusion process in the body is at that speed or slower. Only the fusion of synaptic vesicles is much faster than that. And at each nerve ending, the first vesicle that fuses and releases neurotransmitter succeeds in doing that in about 200 microseconds. Massively faster. And the last vesicle that fuses to release the last little bit of neurotransmitter in connection to this single electrical impulse is within 1 millisecond. 0.2 milliseconds to 1 millisecond. So this is 100 to 1,000 times faster than every other fusion process in the body. Now, there are a couple interesting things about the synaptic vesicle. One is its size. It's a 35 nanometer diameter single membrane vesicle. Every other vesicle in the cell, everywhere in your body, in the cytoplasm, I should say, is about 70 nanometers or larger. But this is by far a smaller vesicle. And probably the speed is somewhat influenced by the way membrane stresses are distributed on a small vesicle compared to a larger vesicle. But by far the greatest mystery is that the very same protein machinery called snare proteins, and I'll give you more details in a moment, the very same proteins are utilized, for example, to release insulin on one time scale and to release neurotransmitter on a completely different, much faster time scale. How can that be? How can you use the same machinery according to a common principle on timescales that differ by a factor of 1,000? So in some way or other, the answer probably, in a very general way, is cooperativity in some way, bringing together many elements that are similar so that a few can succeed faster at a given site. But we don't really know the answer, and that's the problem I'm working on, and that I'd like to talk about today. In essence, how can this process be so fast? And by the way, it needs to be that fast. So if you want to think of our brain as a computer, it has a CPU speed. And that cycle time is probably about 40 hertz, 30, 40 hertz. And you kind of know that because if you see, watch an old-fashioned, as they say, motion picture, before we had digital video clips, they'd be going 30, 40 frames per second, and that's kind of, our brain kind of scrambles that into a continuum, okay? So we kind of know that overall, that our CPU is working at sort of that speed. It's a very rough index. And you have many synapses that need to be passed through to do a computation of that sort. And when you kind of go through the numbers roughly, you're down to a few milliseconds are required, are needed to get through every synapse. That's about what synaptic transmission takes. And if any one of the several steps involved were slower by a factor of 2 or 3, our CPUs would be even slower than they are. And we'd be even more crippled as a species than we are. I'd hate to think about what that would mean. Possibly Donald Trump has found out how to do that. Okay, So that's kind of the general background. Now this is to be a little bit more concrete, and I assume that one of these is the button that you push? Yes. Okay, so this is a nerve ending. This happens to be a muscle cell rather than nerve. So this is a nerve that's impinging on a muscle to cause it to contract. Here are synaptic vesicles, one after another after another. Each one contains many, many thousands of neurotransmitter molecules packed within it. And this is the resting nerve. Now when the action potential comes down the nerve, it spreads, the electrical impulse spreads, and calcium channels, which are in the membrane, very local to these vesicles, open up, voltage-gated calcium channels, and they emit calcium ions from the extracellular space here. This is the so-called synaptic cleft, presynaptic membrane, postsynaptic membrane. Calcium enters as a consequence of the action potential, and within 0.2 milliseconds of the entry of those first calcium ions, the vesicles begin to fuse, and they complete their fusion, as I said, within a millisecond. And as they fuse, their inside becomes continuous with the outside of the cell, and the neurotransmitter is released. It diffuses across this very short synaptic cleft, and on the other side, the postsynaptic membrane, it is teeming with receptors for the neurotransmitter that will open up themselves channels in the postsynaptic cell membrane to emit ions that will then cause the membrane here to be depolarized in general, or polarized depending on whether it's excitatory or inhibitory. And then a computation can take place in the postsynaptic cell. Okay, so that's the anatomy. Now, the snare proteins that I've referred to are the cell's engines for membrane fusion. And they are a family of membrane proteins. They are found throughout the cell and in virtually every cell in your body. And there are many versions of them that are homologous to each other, encoded in the genome. There's a particular set of snare proteins that mediate release of neurotransmitters, and also, by the way, the release of insulin. As I mentioned, they're the same ones on the two different timescales. Now, in general, there are two types of snare proteins as we categorize them. There are V snares and T snares. V snares reside with membrane anchors, membrane helices. They are anchored into transport vesicles, hence we call them V for vesicle snares. So, therefore, the synaptic vesicle has a V snare. I will refer to the name of that V snare from time to time. It's called VAMP, or it's called synaptobrevin. It has different names, VAMP or synaptobrevin, but it's the V snare of the synaptic vesicle. And structurally, what it consists of is a single transmembrane helix and a second helical domain. It's a little more complicated, but not much more than that. So it's essentially a simple helical protein. Now, each target membrane also has its own set of snare proteins. And these also form helical proteins. And we call them T snares for target membrane snares. So a vesicle is always programmed to fuse with a target. A vesicle that forms, for example, from the endoplasmic reticulum, in general, is targeted to fuse with the next organelle in the secretory pathway, the Golgi apparatus. So it will have its version of the V snare, and the Golgi apparatus at the entry site will have its version of a T snare. In this case, the synaptic vesicle has, as I've said, its own V snare called VAMP, and the T snare in this case, the target membrane is the plasma membrane, the presynaptic plasma membrane, and the the T snare is composed of two proteins called syntaxin and SNAP25, and I will mention their names again several times, but we won't be relying on the names so much. Now, the T snare is always composed of three alpha helices. These three alpha helices can assemble with each other in the absence of the V snare. So that's why we call that entire object the T snare. The V snare, though it's shown as a helix here, when it's by itself is unfolded. So actually, the synaptic vesicle, as it were, has many copies of the V snare, about 70 of them actually, for every small 35 nanometer vesicle. And the cytoplasmic domains, the coil-forming domain, it's actually a random coil. And it's only when the V snare comes in contact with the T snare that it then is templated to fold into a helix. We call that process zippering. And what results is what the protein structural biologists call a 4-helix bundle. And that means 4 helices that are interdigitated into each other through a series of heptad repeated amino acid sequences which are hydrophobic. Actually first discovered in Cambridge by Francis Crick many, many years ago in the 1950s. It's also the basis of forming hair, for those of us who have any. So the beauty of this system is that the snare complex, when it assembles between two membranes, and we call this a snare pin, and you see why we call it a snare pin? Because as the V snare assembles on the T snare and zippers, it pins the synaptic vesicle very closely, in fact within, by definition, molecular contact distance with the plasma membrane. Now as this zippering process proceeds and the V snare is converted from a random coil into a much shorter alpha helix, a force is created that literally pulls the vesicle into the membrane. And that force is sufficient to create, and I won't go into the details here, enough pressure to literally dissolve these bilayers and push the one into the other. The estimates of pressure that we've made from various sources, including direct measurements of the force that is involved in zippering with single molecule measurements by optical tweezers, is probably in the range of 100 times atmospheric pressure, maybe more, right in this very region due to this inward directed force. This fully assembled 4-helix bundle is extremely stable. It's the amount of energy has been measured by any number of methods now, is about 80 kT that stabilizes this, so it's irreversibly assembled for all practical purposes. In fact, the very first component of this machinery that we discovered in the mid to late 1980s is an ATPase that recognizes these fully assembled snare complexes but ignores these snare pins, and which hydrolyzes ATP and uses the energy to literally melt the 4-helix bundle and to allow the snares to be separated so they can be used over and over again. So what I'm really describing to you is a mechanism in which protein folding is coupled thermodynamically to membrane fusion. The T snare is, before fusion, is partially assembled, it's not completely assembled. The V snare is unfolded, and if these proteins encounter each other in solution, they will release about 70 kT as heat. But now, since they embedded in membranes, I think you can see that perhaps they can partially assemble, but there's no way they can fully assemble, and by the way, they are zippered from one end to the other, right into and through the membrane. There's no way that that can happen unless the two membranes are one. So there is a steric mechanism, at the very least, that thermodynamically links protein folding, high energy, to folded protein, low energy, and that energy, rather than being released all as heat, is channeled in one or another way to do work on the lipid bilayers to force them to fuse. Now there's a lot of discussion about what the transition states are involved and how exactly the lipids are rearranged, but at a certain level I think you can see there's a very elegant understanding of this process. And these proteins are re-energized at the expense of ATP hydrolysis when they are melted after fusion and after they've assembled into this so-called cis snare complex, because it's coming from the same membrane, and then unfolded again for another round. So that's the underlying biophysics and biochemistry. Now, timing, a single snare complex is capable of fusion anywhere between 10 and 100 milliseconds after docking. We don't have absolutely precise numbers. There are some measurements that suggest it might be several hundred milliseconds, and there are a range of different assays, but it's somewhere in that ballpark. Again, much slower than 0.1 milliseconds. So back to timescale. The snare complex by itself in isolated membranes, even synthetic lipid bilayers, is capable of completing fusion in about 10 to 100 milliseconds. 100 1,000 times roughly slower than the synaptic vesicle fuses. And by the way, even a single snare pin is sufficient for bilayer fusion. And this has been replicated by many labs. If you have a single snare pin, if you give it enough time, it will fuse. That time is about half a second to a second. What I'm describing with these times is actually when you have many copies of VAMP in the vesicle. But when we take a very artificial situation where we have just one molecule of VAMP, then the time it takes for this zippering to be completed is in this range. Still, very much slower than the release of neurotransmitter. But what it points out is that the energy made available by a single snare pin thermodynamically is enough to overcome the activation energy for two membranes to fuse, which Frédéric Poncet and I and a student at École Normale Supérieure have recently directly measured. We've got the first direct measurements of it, and it's between 30 and 40 kT. So a single snare pin is apparently adequate for that. Okay, so that's the background on snare-dependent membrane fusion, and whether we're talking about biosynthetic protein transport, the secretory pathway, endocytosis, you name it, this is the underlying biophysics. And it's only in the case of the synaptic vesicle that fusion is so fast. So part of the answer to the question, how can fusion be so fast, comes from the fact that the vesicles that are docked up against the membrane, I'm going to go back a slide or two, they constitute what's called by physiologists the readily releasable pool. So I have to introduce you to a little bit of physiology. The vesicles you see out here, they're in no position, right, to fuse with that membrane, certainly not within a fraction of a millisecond. But it's only the guys that are lined up down here that are in a position, potentially, to fuse in that very short time range. They constitute what is called the readily releasable pool. And so that means that when you have a single impulse that comes by, there are vesicles that can release their neurotransmitter, and there are, in a typical synapse, this is a neuromuscular junction, which is quite different than a central synapse. But up here, there are probably about 10 on the average vesicles in the readily releasable pool that are docked directly on the membrane. The rest of these essentially are replacements that will move up to the front line after a round of neurotransmitter to replace the ones that fused. When this vesicle becomes that vesicle, the readily releasable pool, that's called priming. And so there's a whole terminology that is used around this, which I'll mostly try to avoid, but just for those of you that have been reading papers in the field, I just wanted to mention that. So then, what is the state of the snare complex in this readily releasable pool? Okay? Well, as it turns out, there are several lines of evidence that establish that in the readily releasable pool, the snare complexes have already begun to zipper, and in fact are about half zippered, which raises an interesting question. If they're half zippered, what prevented them from going the rest of the way? Because the zippering process is in fact entirely downhill energetically. Okay? And that kind of is the meat of what I want to talk about today. So, how do we know that they are approximately half zippered in the readily releasable pool as it's defined by physiology? So these are studies that have been done. Again, there are several lines of evidence, but the one I like the best, I find it just simply the most elegant, comes from Charlton at the University of Toronto. And what he takes advantage of are the particular properties of botulinum and tetanus toxins. Okay? Now, I know everybody here has heard of tetanus and botulism, but maybe not necessarily in this connection. Okay? So these are bacterial toxins secreted by typically anaerobic bacteria that cause the disease that we call tetanus or botulism. And what happens really in these cases is that muscles are paralyzed or in rigor, and as a result of the fact that synaptic transmission comes to a halt. And over a period of several decades, the classic Cesare Montecucco at the University of Padua in Italy solved the problem of how these toxins cause paralysis. He discovered they are proteolytic enzymes, and they have a subunit that binds them to a nerve cell, and it engineers the entry of the protease portion into the cytoplasm of the nerve. And these proteases are extremely specific and potent. And in fact, each one cleaves just one protein in the cell or in the brain. And that one protein is in fact a snare protein. Several of these botulinum species and tetanus toxins address the V-snare vamp synaptobrevin. Other ones which are not indicated here target SNAP25 or syntaxin. So this actually, early on when we discovered the snare complex, was a very important line of evidence that helped us appreciate quickly the physiological relevance of the biochemistry. Okay, so you can see very clearly, I think, that if you whack-a-mole at these proteins, you're going to stop synaptic transmission. But fortunately, it's a little more specific than that. So for example, botulinum, these two toxins recognize this amino acid sequence. This one recognizes that amino acid sequence, but interestingly, they cleave a different bond. So in other words, the sequence here in VAMP is recognized, is glommed onto by this proteolytic enzyme, which is positioned to actually cut right here. Here's another botulinum toxin, which recognizes this sequence, okay, and cuts the same bond. Okay, isn't that kind of interesting? Okay. Now, why is all that relevant to what I'm telling you? It's relevant because when this sequence is sequestered in the 4-helix bundle, it can no longer be recognized because it's buried. So if you have a snare complex, the recognition sequence is zippered in the readily releasable pool, the readily releasable pool will be immune to the toxin. On the other hand, if in the very same snare complex you have a more membrane proximal sequence that is not yet zippered, then the toxin that recognizes that one will cut the very same bond. Does that make sense? And this is exactly the experiment that Charlton has done in a variety of different synapses. And it turns out that if you recognize this sequence, the toxin cannot cut or inhibit the release of the readily releasable pool. But if you recognize a sequence that zippers in the second half of VAMP later, the one that really completes the fusion, well then you immediately shut down the readily releasable pool. So that's the type of experiment that really, I think, very convincingly shows that the snare complex is somehow frozen in a half-zippered state, approximately. And so part of the answer of how neurotransmission can be so fast is that the snare complex is half-zippered, okay? Now you might say, well, why isn't that the whole answer? Because if you're half-zippered, you just got to release it, and then kaboom, it fuses. The reason is that 10 to 100 milliseconds that I quoted before, okay, when you have a bunch of snares, that actually is the time to completion after half-zippering. So this is probably good for something, probably a lot, but it raises yet another question, which is what is it that's holding the snare complexes back from fusing? Okay? Now, happily, we know a lot about the machinery that regulates the snare complex at the synapse, at the presynaptic membrane. But we don't yet understand exactly how that relates to this process that I just described, freezing the snare complex. And that is what I will come to momentarily. So what are the questions then that I'd like to address in the next 30 minutes? Okay, the first question is, because the half-zippered state is intrinsically unstable, it raises the question, how is this state stabilized in the active zone at nerve endings? And again, the second question is, since snare proteins generally take hundreds of milliseconds to complete their fusion from this state, how can the release of neurotransmitters be released so many orders of magnitude faster? Okay, so those are the core questions that I hope to at least begin to address with some recent experiments. Now, I alluded to the fact that we know a lot about the machinery that is important in regulating membrane fusion. So what I've been describing up until now is the snare complex. And as I said, one or another version of the snare complex is needed for every membrane fusion process, virtually every membrane fusion process in the cell. And a particular snare complex, VAMP, syntaxin, SNAP25, is used at the synapse. There's another equally ubiquitous protein called the SM protein, discovered by Suthoff and independently by others. And for every snare complex, there is a unique SM protein. And this protein is a privileged chaperone. We don't yet understand all the details about how it works, but its job is to help the N-terminal domain of the snare complex assemble to get it to this state. And although the snares will assemble spontaneously, the speed with which they will do that is dramatically increased by that specialized chaperone, which binds to one end of one of the snares so that it's locally concentrated on the T snare, so that when the V snare comes into range, it can help assemble these helices in the right orientation. That's essentially what it's doing, is orienting helices. But we, again, don't know as much as we'd like. Now, this protein, in the case of the synapse, in the case of the synapse, this protein has exerted its action primarily to get to the half-zippered state, right? So I just want to get it out. I got it in the picture, now I want to get it out of the picture. Now at that point, that last, from the readily releasable pool for rapid release, well, that involves other machinery. And the two key elements of that machinery are synaptotagmin, the calcium sensor, and complexin, another key regulatory molecule. And I will briefly mention complexin, but I will then now spend the rest of the talk talking about synaptotagmin, because that's what our most recent data concerns. Both of these proteins were discovered by Tom Sudhoff with colleagues many years ago. Tom especially, of a special note, established that this protein, synaptotagmin, is the key sensor for calcium. So you remember I mentioned that when the action potential comes to the ending of the nerve, voltage-gated calcium channels open, calcium is admitted. So obviously then, there has to be a protein or something that binds calcium and says, okay, calcium's here, now let's release the neurotransmitter. And for what I've told you so far, you can imagine, since synaptotagmin is that protein, that synaptotagmin, but only when it binds calcium, must interact in some way with the snare complex and or whatever is freezing it, preventing it to go from completion, from the half-zippered state to completion. Hope that's clear. Question, how? We don't know. Okay? So that's really, in a way, what we're trying to figure out. Now, a bit more about the anatomy of synaptotagmin. It sits in the synaptic vesicle. It has two very similar domains called A and B, each of which is capable of binding several calcium ions. And so especially it is the B site which, when it binds 3 or so calcium ions, triggers neurotransmission. That's the key event. And that was Tom Sudhoff's discovery that really nailed down the role of this protein. Complexin is a relatively simple helical protein that lies on the outside surface of the 4-helix bundle of the snare complex. So in other words, it's a helical regulator of a 4-helix bundle. Now what does that protein do? And what happens when you take one or these other proteins if you delete them from the nerve? Okay? If you delete synaptotagmin from a nerve, there's no neurotransmitter release. So it's just simply obligatory. It works actually at several stages, but it works from the very earliest stages. And so if you take it out, nothing happens. If you take out complexin completely, it's really interesting what happens, and what doesn't happen. The first thing that you notice is you still have synaptic transmission, but just not as much. So what the physiologists call evoked potential, which is just a measure of how many vesicles fused and got their transmitter to the other side, goes down by about half to 2/3, depends on the system and so on. So calcium-dependent neurotransmitter release and the readily releasable pool, so to speak, still exist. But there's a difference. It's no longer synchronous transmission. In most systems, the synaptic vesicles, they may start quickly, but they dribble out over the better part of a second. So that wave of neurotransmission that would have been compressed into that 1 millisecond, okay, is now dribbling out it's what's called asynchronous neurotransmission. So it's coming out over the better part of a second. More than that, even, is spontaneous release. Now, like every system, if it's finely tuned, has a certain rate in which an event happens by itself when you don't want it to happen necessarily. Okay? So in the absence of an action potential, there's a certain frequency of release of individual vesicles. It was actually, that was the key discovery of Bernard Katz in the 1950s that led to the recognition of the role of synaptic vesicles, and it's why it's called quantal transmission, because individual quanta would fuse. And he discovered that, that an entire round of neurotransmission is the summation of many individual quanta. And that was a very important advance in understanding neurotransmission. The release of the spontaneous release, when you take away complexin, goes way up. For example, in some organisms, it goes up over a factor of 100. So complexin is doing two things that are important. It's basically, in my mind, the intelligence protein. Okay, it's a dumb little helix, okay? But can you imagine how blurry the world would be, okay, if every time you wanted to think a thought, it came spread out over a second? Okay, I mean, you know, I'd be watching you nodding your head or talking in your ear. If you listen, I'd have to have lip syncing. I mean, it would all come out as a complete blur. So synchronicity, okay, and the noise level, think about that. So the noise level goes up and synchronicity goes down. I mean, you're really dumb, okay? You may survive, but you're really going to be dumb, okay? So complexin is a very, very important protein. But it cannot be as important as synaptotagmin, because at least you get calcium-dependent release with synaptotagmin. And if you take away synaptotagmin, you got nothing. So we're working actually on both proteins, but because they're both important. But complexin, in a broad sense, has, at least the way I think about it is, the core system that involves freezing the snare complex and triggering a release with calcium is somehow synaptotagmin. I'll show you how in a minute. And that the system is further energized by binding complexin, and that cooperativity is somehow further increased so as to produce a more, you know, if it were a higher energy or strained system from which release can be more explosive. I'll put it in physical terms because I tend to think about it that way. Good, so synaptotagmin. And only one more bit about synaptotagmin background before I can show you some recent experiments, because it is in fact a bit of a complicated protein. A domain, B domain, each binds calcium. There's another patch up here that binds to a phospholipid called phosphatidylinositol bisphosphate, PIP2. Now, PIP2 is a negatively charged phospholipid that is found on the inner surface of the plasma membrane, and in particular at the inner surface of the presynaptic plasma membrane at the active zone. And you remember I said a moment ago that synaptotagmin is needed even from the very beginning? Well, from the very beginning means to actually dock the vesicle close to the membrane. Even before the snares have engaged, the synaptotagmin, it appears, needs to bind to PIP2. And when synaptotagmin emanating from a synaptic vesicle reaches across and binds the plasma membrane by binding PIP2, now the vesicle is stuck on the membrane. Okay? If you do away with PIP2 in vitro systems, or you mutate the specific binding site here, which is also called the polybasic region, I'll be making reference to it in a couple moments, you eliminate binding to PIP2, and you eliminate the formation of the readily releasable pool and the very closely morphologically docked vesicles as seen by electron microscopy. Okay? So that's an important bit of background. And one more last bit of background concerns what happens to this protein when calcium binds to it. Okay? Now, in general, when you have signaling processes, when a ligand binds to a receptor, there's two general mechanisms that are involved in transducing a signal, as I'm sure you probably know. One mechanism is the protein changes shape. That's allostery, as first discovered by Changeux and colleagues. Okay? The other mechanism is you don't change your shape, in which case, what happened? Well, you have to change location. Okay? And that probably is the most important signaling mechanism in eukaryotic biology. Your localization changes, and therefore you create proximity relationships that would otherwise not exist. It is the latter mechanism that synaptotagmin uses. but it's a very local topological rearrangement. Jose Rizzo in Dallas, for example, has captured the X-ray structures of this domain or this molecule with or without calcium. They are exactly the same. There is no conformational change. So the only thing that can be different is the positioning. And that position is drastically different. Actually, synaptotagmin's B domain and its A domain each insert into the lipid bilayer. So when calcium binds, they literally insert certain aliphatic amino acid side chains into the bilayer. Now, in order for that to happen, of course, you have to push the lipids out of the way. There's a high activation energy and therefore threshold for that to happen. And what provides that activation energy? It's the binding of calcium. How? Calcium actually forms a bridge between the protein and an additional group of acidic phospholipids. So just to avoid confusion, there are two, it is confusing, there's acidic phospholipids are used at two stages in this reaction. In the first stage, specifically PIP2 is used to dock the vesicle to help trigger snare assembly, half zippering, and the readily releasable pool. That happens in the absence of calcium. Now, calcium comes in, voltage-gated calcium channel opens, calcium binds to synaptotagmin, and it binds right here, in fact, to synaptotagmin, to aspartic acid, negatively charged residues. And it binds not just to these residues, but it binds to the head groups of other negatively charged acidic phospholipids in the plasma membrane, either phosphatidylserine or phosphatidylinositol bisphosphate, mostly phosphatidylserine, which is much more abundant in this case. And so, again, let's go over it again. Here, in more detail, is the positively charged patch, the polybasic region of synaptotagmin's B domain, bound to PIP2. So that's synaptotagmin. This is the first interaction that docks the vesicle, positions synaptotagmin on the surface of the plasma membrane. It now needs to rotate 90 degrees, but that's it, in order to form a calcium sandwich between acidic residues on the protein and acidic phospholipids. The geometry of that sandwich, as discovered by Ed Chapman a number of years ago, results in the insertion of these aliphatic residues. That is the molecular switch that releases the neurotransmitter somehow. We know that because you can play with the negative lipids and you can play with these amino acid residues that bind calcium, and that switch is toggled by that interaction. Okay? So now you know everything that we knew going into this study. And what I'm going to tell you is that of a new discovery that my colleagues and I at Yale have made, which is that synaptotagmin actually forms a polymeric ring. These AB domains emanating from the synaptic vesicle polymerize with each other into a closed circle. The circle is variable in diameter. It can have 15 to 20 in that range synaptotagmin subunits. These synaptotagmin rings, as we refer to them, are based on the B domain. So, and I'll show you a structure of it. So it's basically the C2B domain polymerized B around in a circle. From everything we know, we hypothesize, though we have not proven yet directly, that this ring forms at the interface of the synaptic vesicle and the plasma membrane, and it in fact forms when the B domain contacts PIP2. As you'll see in a moment, binding to PIP2 triggers a C2B protomer to form a ring. And that ring dissociates when calcium binds to the B domain. And I'll show you structurally why that works, how that works. But the big idea is, if you want to prevent a vesicle from fusing with a membrane, what could be simpler than simply putting a wall in between? And if that barrier goes away when calcium comes, then fusion can proceed. Now, I don't think it actually is that simple. In fact, I can guarantee you it's not that simple, because nothing in biology ever is. But that is the core underlying concept I believe to be the case. Why would this freeze the snare complex in a half-zippered state? Well, it turns out that, that the height of this ring, which is the height of a B subunit, is about 4 nanometers. 4.5 nanometers. The snare complex, when it's half-zippered, is able to form across a 5 to 10 nanometer gap. But it cannot zipper beyond that. And there are 2 more facts that I need to get out that come from related studies in our lab. With Frédéric Poncet and his colleagues at École Normale Supérieure, we've used the surface forces apparatus to measure the gap at which the half-zippered snare complex forms. And it will form, as I've said, between 8 and 10 nanometers separation between 2 bilayers. So we know that this half-zippered snare complex can form. We also know from work with optical tweezers that we've published with Yongli Sang and his colleagues in my department at Yale, where we actually pry apart the last part of the snare complex to zipper with 2 beads and toggle them back and forth. We know that they toggle all or none like a binary switch. So you're either half-zippered or you're fully zippered, and this thing can't get past half-zippered because it can't get close enough to be fully zippered because of the spacer. So it's probably not that simple, but that's the core concept that we have. Now, what is the evidence that supports that? Well, here what we've done, and this is the work of a postdoctoral fellow, Jing Wang. What she has done is take pure, expressed and purified synaptotagmin cytoplasmic domain, AB, just that protein, nothing else in the test tube. And she now adds to that PIP2. Now that PIP2, it normally will form a big micelle or a or a vesicle, but we've used a short chain analog that has only 4 carbons instead of the usual 16 fatty acid chains. So it's actually soluble as an individual monomer at the concentrations that we use. And with a binding constant of a few micromolar, it triggers the polymerization into rings, such as you see here. These rings can be separated by centrifugation and purified. They remain intact. They can be monitored quantitatively by fluorescence correlation spectroscopy as they move by. We can estimate by that method that they have about 15 to 20 subunits. That's exactly what, knowing the size of a synaptotagmin, there would be one, there would be another, there would be another. We could imagine how many might form a ring of this circumference when the diameter is typically 25 to 35 nanometer diameter. So there's about 15 to 20 of these synaptotagmins, and this protein by itself can form a ring. Okay, so there's no doubt that this protein has the capability of forming a ring. The ring will not form if we mutate the polybasic region of synaptotagmin, which we know binds to PIP2. I don't have the data to show you today, but the B domain by itself will form a ring. What happens if you enable the ring to form on the surface of a membrane? So here we take lipid monolayers. Again, this is negative stain electron microscopy. Same experiment as before, except now the PIP2 is on a proper PIP2 molecule that's inserted into the lipid monolayer. The protein is added above the monolayer. We rinse, and we look to see what do we see on the surface of the monolayer. Well, you actually see lots of rings, okay? And those rings are the same diameter as those that form intrinsically in solution. However, something is different about the rings on the membranes. You see the uniform electron density? This is an empty ring that's simply deposited on an EM grid. These are different. You see this, that the stain is excluded by something that's bulging out above the ring. What's happening here is the lipid monolayer is actually being sucked or puckered into the inside of the ring. Okay? So there's a very tight interaction with the PIP2 in the membrane. These rings, of course, don't form. These form in the absence of calcium, and they require PIP2 or an acidic phospholipid, and they require the polybasic region. I want to make one other correlation. I've told you already there are 70 or so vamp V-snares in every synaptic vesicle. There are 15 to 20 synaptotagments in every synaptic vesicle. So the number of synaptotagments in a vesicle is ample. In fact, seems to be just right to form rings of about this size, about 30 nanometers size, outer diameter. Remember now, the vesicle is 35 to 40 nanometers in size. So this ring is a substantial portion of the diameter of the synaptic vesicle, but it still is within the confines of the synaptic vesicle, so to speak, in the shadow that is cast. Now, happily, sometimes the ring will go to close, but it doesn't actually close. It actually kind of gets bumped up a notch. And that causes it to continue to polymerize, and it actually polymerizes into a helical array, which continues to suck the membrane into it, just like when you have a single dome coming out of a monolayer. And the result of that is a membrane bilayer tube, which has a protein array on the surface. And oftentimes these arrays are organized well enough to diffract. And that's what you see here, and we can then use standard methodologies to reconstruct from negative stain and cryo images what the packing in these arrays is, which is presumably very close to the packing in a closed circle. And that's what's shown here. And so what Jing Wang, again, who's responsible for most of this work, has done, here's a tube, okay, here it is in cross-section, here's the cross-section view, here's the cross-section view, here you're sectioning, along, you're perpendicular to the tube, and here along the length of the tube. So a ring, looking down in this view, would be the equivalent of looking down on a small portion of a ring. And what you see as we fit the electron density is the B domain is up against the membrane, the A domain is pendant. And from reconstructing many rings, we know that the B domain is in a standard geometry of about 15 or so, depends on the helical pitch. We can get anywhere, tubes from 15 to 20 around, okay? And it's always the B domain that's up against the membrane. The A domains are pendant radially. And though in any one tube they may be in a given geometry, from tube to tube they vary a lot. So our concept is that the ring consists of B, 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 B. A is radially and more or less random in the absence of any other component or factor. Now, the polybasic region shown here is in immediate contact with the membrane. And if you look at a side view of the ring, which means this view, this is what the membrane would look like. And so now you can see why there might be a dome, because the way to picture this is here is the ring kind of inside. The ring is now coming, this is the bilayer or the monolayer in the experiment, and the ring is going like so, okay? And the dome that you see is essentially this membrane being sucked at or above the ring, okay? And now you might ask, where on the surface of this ring, according to this structure, is the site that binds PIP2? The answer is it's on its inner surface. So if you picture a tire, like a car tire, it is lined on the inside with these polybasic regions. So it's perfectly understandable now why the membrane is puckered like that. And by the way, going back to the early 1950s, the great cell biologist, really the founder of cell biology, George Palade, noted that the membrane, the plasma membrane, in cases of regulated exocytosis, is always puckered. So it may very well be that that pucker has its basis in this. We don't know that, of course, but it might very well be. Now, very interesting to ask, where is the calcium binding site? Now, I want to emphasize this structure forms in the absence of calcium, right? But let's pretend that calcium was there. Where would it be? It would be right here. So that's kind of interesting, isn't it? Because actually the calcium binding site is in the region that seems to be the contact region between the two B domains. I'll show you in a moment that it actually is. Okay? So what will happen to this ring according to this geometry? When calcium binds to this site, what will happen? Well, the nearby region is where this loop is, this aliphatic loop that needs to insert into the bilayer. How can that happen? Remember, a 90-degree rotation is required, and in this case, a translocation of perhaps 20 angstroms. So, just looking at this, you can see that the existence of this ring is incompatible with binding calcium, so that you would predict that when calcium binds to this site, the subunit contact, the BB contact, will be broken up. The synaptotagmin will have to rotate into a topology completely incompatible with the domain, because the interface between B and B is now the same interface that's on the membrane. Make sense? So is that true? So this is what happens when you preform rings and you add calcium. And in the relevant range, 0.1 to 0.5 millimolar for local calcium, you see that the rings literally disappear. Interestingly, by the way, the soluble rings are completely resistant to calcium because they have no alternative conformer to flip to, and that just points to the role of a membrane insertion in stabilizing the state that triggers exocytosis. Now, what happens if we mutate residues involved in calcium binding, such as the aspartic acids? Well, you should no longer see, if this is right, the ring should be stable and immune to calcium, and that's exactly what happens. So this is one of several lines of genetics that connects the ring to the behavior of the ring to the behavior of exocytosis. You mutate the calcium. Calcium is required to dissociate the ring. The ring assembles in the absence of calcium. The ring is triggered to form only under the very condition that docks the vesicle binding PIP2, requiring the very same site that genetically is required in a living cell for docking to occur, and then therefore for snare engagement to occur. So there's a lot of reasons to begin to believe that this ring is an important physiological actor. Interestingly, the rings that actually are more stable when you mutate calcium binding than they might be otherwise, and they're actually more stable in a calcium-dependent way. And what we think this is due to is the A domain. Because even though we've removed calcium binding by the B domain, the A domain is still there, and that A domain will now insert on the outside of the rings and probably further stabilize the ring. So we think that's what's going on, but we haven't explored that further. Now, we don't have a high-resolution structure yet of this interface, but we do have some good idea of what the contact region might be. So Oscar Bello in the lab has systematically mutated the residues predicted to be in that region to alanines and to other amino acids. And the idea is that if we're lucky and we're right, we should hit some residues that are actually involved in the B2B contact, and when we do, the ring shouldn't form. And maybe we'll be lucky, we'll get some mutations that won't affect, in which the B domain can properly fold, synaptotagmin can properly fold and function, and maybe those won't form rings, maybe even in living cells, and maybe then we'll be able to test our hypotheses about more specifically in a physiological setting. And so he systematically mutated all of those residues and tested the properties of these synaptotagmins, and maybe half of them have no effect, but half of the sites definitely do. And what they do is they eliminate ring formation, really very profoundly. And interestingly enough, and I won't show you this data, they do so in a dominant fashion. So if we take a bad synaptotagmin B domain and we put it in with a good one, what would you expect would happen? You start growing your ring, and then you get a bad one in, and now it can't grow any further. And in fact, that's exactly what happens. And you produce sort of a variety of sort of semicircular C-shaped structures. So you would predict, therefore, that a mutant in this binding site that doesn't form rings should dominantly interfere with synaptic transmission in a genetic sense. If you take, normally in genetics, you have to have two bad genes to have a phenotype. But if you have a dominant gene, if you have a multimeric machine, and you throw one bad piece of machinery in with the rest, it screws everything up, meaning that they're cooperative. Okay? Well, nature actually did this experiment for us. This is a family of people that appeared in a clinic because they had neuromuscular weakness, and that was intermittent, and a number of other relatively nonspecific signs that suggested synaptic disease. And I have to hasten to mention that the genome encodes something on the order of 20 different synaptotagments. So don't think that there's just one synaptotagment. Synaptotagment 1, 2, and 9 are the principal sensors for synchronous neurotransmission, and other synaptotagments are involved in calcium-regulated exocytosis of other kinds. Okay? Synaptotagment 2 is particularly concentrated, but not the only synaptotagment at the neuromuscular junction. So let's imagine that this mutation might be blocking ring formation. Would we expect to have an absolutely complete blockage? Probably not, because that person would be dead. We would expect to see, if there's any mutation at all, a so-called weak allele, which has kind of a partial effect. And as it happens, this mutation is in residue 310. That happens to be the very same residue. This is what the people have. The family has this has a mutation of P proline in amino position 310 of synaptotagmin 2, changed. In this case, we put it in a leucine, but in the family, it's actually an asparagine, and that mutation is not as strong. There is still some rings that form, but nevertheless, it's a very striking fact. So what this demonstrates then is the physiological importance in the human body of this very same residue, quite possibly for the reason that it doesn't form rings. And I do want to mention that when Oscar Bello characterized these, he showed that each of these synaptotagments is folded. He showed that it binds calcium with the normal energy and binding constant and kinetics, and that it binds that they bind PIP2 by the basic region in the normal way, and that they insert their loops in a calcium-dependent way. They insert into the bilayers in a normal way. So everything else about these synaptotagments is normal. So then you can see why we're beginning to think that a ring of synaptotagmin may form when synaptotagmin encounters the plasma membrane, that enables a snare complex to zipper up to about halfway, and that when calcium binds to this as a result of triggering neurotransmission, the ring is the ring is broken, and in one or another way, the snares can complete their exocytosis. We would also predict from this model, and predict very strongly, that in the absence that a synaptotagmin that could not form this ring, but which was normal in every other respect, should not provide this impediment for fusion, and therefore the rate of spontaneous neurotransmitter release and the absence of action potential should go up. Does everybody follow that? Because there's no impediment. The synaptotagmin may be there, but it's not in the form of a ring. So that's a very strong prediction. And most recently, another postdoctoral fellow, Warden Jouanneau, has begun to test this, not in nerves. We're working our way up to that at University College London in collaboration there in hippocampal cultures. We don't have that data yet. But when we transfect in a PC12 cell, which is a mimetic, a cellular mimetic, we put in the wild-type gene, we observe the same rate of spontaneous fusion. But when you put in this mutant that doesn't form rings, but is otherwise normal, you get a drastically increased amount of spontaneous release, exactly as you'd predict. And by the way, this mutation is dominant over the wild type. Now, the docking of the vesicles, we measure actually the number of vesicles docked, is unaffected. So the docking of the vesicles, as measured by turf microscopy, which is the morphological equivalent of the readily releasable pool in this system, is the same, but the spontaneous release rate is way higher. Okay? So this is a very strong prediction that's tested, and now in preliminary data at least is confirmed. So we don't know whether, I mean, to say what do we know and what we don't know, there's no evidence against this hypothesis. It's a very simple hypothesis. There's a lot going for it. But we have yet to visualize the actual ring of synaptotagmin at an actual interface between an actual synaptic vesicle and an actual presynaptic plasma membrane in an actual nerve. And we're working our way up technically to doing that, but we don't have that evidence. And so in a way, we don't have a shred of morphological evidence that this ring exists, except that the physical chemistry says that it should, and so we operate under the assumption that it does. Now, are these ring-like oligomers a general feature of synaptotagments? Well, actually, as I've mentioned, there are many different synaptotagments in the body. Synaptotagmin 1, 2, these are synchronous sensors. Synaptotagmin 7 is widely believed to be an asynchronous sensor. It mediates a separate process of synaptic vesicle release, which occurs on a different time scale. It also forms rings. This is the work of Natalia in our lab. Another non-synchronous asynchronous sensor, DOC2B, also forms rings. And in collaboration with Pietro de Camilli in his lab at Yale, most interestingly, a synaptotagmin member of an extended synaptotagmin family forms rings as well. And what these proteins are, these are synaptotagmins that are not involved in exocytosis. It's actually very interesting from an evolutionary point of view. These proteins are found in yeast, which do not have calcium-triggered or any other form of regulated membrane fusion, regulated exocytosis. And what do they do in yeast, and what do they do in our cells? They form adhesions between the endoplasmic reticulum and the plasma membrane, so-called cortical endoplasmic reticulum, which brings the endoplasmic reticulum stably up against and glues it to the plasma membrane and glues it reversibly. Okay? Typically, calcium, when it enters, causes these adhesions to break up. It's not the only method, but typically. And it specifically allows these contacts to form reversibly and without membrane fusion. The whole purpose of an extended synaptotagmin, which is quite a different protein, except it has similar C2 domains within it, its purpose is not membrane fusion. It's to allow two membranes to come within molecular contact distance without membrane fusion, and to do so reversibly. And why? Because it allows, for example, lipid exchange mediated by transfer proteins that are part of that protein. It allows other materials to be exchanged between membranes without the membranes losing their identity by membrane fusion. Okay? So there's a very interesting evolutionary concept that I would like to just introduce, and which can point to how the process of regulated exocytosis, the release of insulin, and in the most refined fashion, the release of neurotransmitter, how this could have evolved. As I said, yeast, for example, have snare pins. They have a synaptotagmin that can form rings, though not a vesicle-bound synaptotagmin. And the yeast snare pins, like every snare pin, their job is to irreversibly, remember that very strong binding energy, irreversibly adhere membranes, causing fusion as a result of this irreversible adhesion. Synaptotagmin rings, I would suggest, have as their basic purpose the reversible adhesion of membranes, but preventing fusion. You can easily see how the coming together at a certain stage of these two principally separate mechanisms could easily come together in one or another way simply by adding binding of one to the other, and then you would have regulated exocytosis in a single evolutionary leap. And I suspect that's probably what happened. And what that suggests, in a way, the idea that there are two separate mechanisms that came together, suggests actually that, contrary to the way that I've drawn it, the snare complex may actually interact with synaptotagmins. I'll probably say a word about that. Do I have another 5 minutes or less? Yeah. Is that okay? Okay, so what then, science, the nature of science is you try to answer some questions, you actually generally fail, right? So I've generally failed. I haven't answered the questions I posed to you completely, but I think we made some progress, okay? And as a result, new questions remained. Some of them are reshaped from the original, from what I started the seminar with, but also some new ones. So the questions we started with. The half zipper is intrinsically unstable. How is this stabilized in neurons? We suggest that the core principle that stabilizes them is the synaptotagmin ring. So we think, we haven't completely proven it yet, but we think it's very likely that this is an integral part of the answer. Snare proteins generally take a leisurely pace to fuse, but it's much faster in the case of neurotransmission, frankly, we haven't answered that one yet. Because, synaptotagmin by itself, well, let's just take insulin release. Insulin release happens when you have synaptotagmin without complexin. It takes, hundreds of milliseconds. Good enough for the pancreas, but no complexin in those cells as far as I know. Only synaptotagmin. So it's a step away from becoming a nerve. Somehow complexin is integral in creating a more energized state. And there are some ideas about that, but I won't take the time now to talk about it. Other problem is simple diffusion. How long does it take for, even if this ring broke up immediately, it would take many milliseconds for these subunits to diffuse away and simply get out of the way. It's probably too slow. Well, it's probably plenty fast for release of insulin, but not fast enough for the release of the neurotransmitter. So I'm assuming that in one or another way, when complexin is added into this mix, it is energizing this membrane to the point that the release is so explosive, and this is the modeling component, that the synaptotagmin ring, under enormous strain, but perhaps due to its symmetry, has balanced forces internally. Now its symmetry is broken, and when the first bond between B and B is broken, and it appears that only one synaptotagmin needs to actually bind calcium. I think there's reasonable agreement on that. When that happens, perhaps there's an explosive release in which the components of the ring are conveyed away as this force is released due to the loss of symmetry in a structure that we've not yet determined. So that's kind of a vague way of thinking about it. Certainly, the snare complex here, I'm showing it ignoring the ring just to be simple-minded. It doesn't ignore the ring. It can't ignore the ring because we know synaptotagmin binds to the snare complex. It binds to form a one-to-one complex. It has several different binding sites. Am I going to show you that? No, I guess I'm not going to show you that today. But Axel Brunger at Stanford has solved that structure, and it's very clear that that the snare complex, or the snare pin that's half zippered, is physically in the mix with the ring. We just don't know how yet, so we're actively engaged in cryomicroscopy and the like to try to figure out how. We have lots of models. They mostly contradict each other, and so I won't show them to you. But we have models. It's not enough constrained by data. The last thing I'd like to show you that comes to and bears on the speed comes from not yet published work in collaboration with Ben O'Shaughnessy's group at Columbia, mostly done by Ben and his colleagues. And I understand that he's going to be coming to give a talk here not too long from now. So, and my personal involvement in this work, although I'm an admirer of it, is relatively peripheral. So, but I want to show it to you because it's in the domain that some of you are interested in. But if you have difficult questions, you should save them for Ben. Okay? But essentially what he and his colleagues have done is to produce a coarse-grained simulation of snare, but a pretty realistic simulation, in which end snare complexes start out between a synaptic-sized vesicle and a planar membrane, and they start out in random orientations. He starts them out in the half-zippered state, and he allows them the ability to zipper residue by residue with a very realistic force function that comes from these optical tweezers that I just described. They have a realistic charge distribution. The plasma membranes have a realistic charge distribution, and as does the synaptic vesicle. And they're using rate constants that come directly from measurements. So as far as I can tell, it's a well-constrained kind of a modeling. And so what their first finding is that, which I'm not showing you here, is that these proteins self-organize into rings. You don't put them in rings, but they self-organize into radial rings, much as you'd expect, as everybody has diagrammed them. And they do it based on entropy. They just keep bumping into each other and excluding each other, and they push each other out. And so they get their best arrangement, their most favorable arrangement, sterically, is in the form of a ring with the transmembranes and therefore the point to which the zippering towards a common center. But because of the entropic factors and collisions, they tend to push each other out radially, which tends to put additional force on the vesicle and creates pressure between the vesicle and the membrane. And that's where some of these pressure simulations, one of the sources, but not the only source of the pressure simulation that I was referring to. And so, then what they do is they model how long does it take for one of these snare complexes, anyone really, to completely zipper, with the idea that they're zippering partially or further completed and so on. And then the first guy that zippers is when they consider the membrane to fuse. So in a way, you have a bunch of these guys, N of them, you let them go, and whoever wins the race first, okay, without any other kind of cooperativity, and there are other kinds of cooperativity that could be possible. And they find that there's an exponential, very clear exponential behavior in which the waiting time, so what do I mean by waiting time? Waiting time is you have your vesicle with N snares, half zippered, and how long, in many trials, does it take for half of those vesicles to have got that first snare complex completely zippered? Hope I'm being clear, okay? And of course, the more snares you have, the less waiting time there is. And this is the result that they get, and it's a very striking result. And so by the time you get to, they only show 10, but it goes up to 15. By the time they get to about, I don't have their most recent data, by the time they get out to 15, it's about 1/1000th of the waiting time at time 0. So what is the waiting time at time 0? That's the time it takes for a vesicle to completely fuse with a single snare complex from the time that that snare complex half zippered. Okay? So you'd think by now that we would know that number. We don't know that number. And the reason is we can see in optical experiments when a vesicle stops moving on a membrane, but we can't actually measure when the N-terminal domain of the snare complex molecularly zippered. I mean, we can, and those experiments are in the works, but we don't know at that point. So we could start our clock at the time when the vesicle comes in contact with the membrane, and when we do, that's where these numbers that come from in the range of half a second or so. Okay? But how much weight there is between there and the time that's half zippered, we actually don't know. Okay? So upper limit, let's say, in the range of half a second. So if you put this number, and you see I'm a little sloppy with the numbers, but if you put that number at half a second and you get up to about 15, you're getting down to the range of a fraction of a millisecond. We don't really know if this is an adequate explanation. It probably isn't, but it's certainly a strong piece of it is simply having N snare complexes, N being a pretty large number, that are held at bay in the half zippered state, and if they're simply released at the same moment, okay, that what this tells us is that all other things being equal, it's possible to get into the right temporal domain with the same physical chemical mechanism at the level of a single snare complex. So I find this not the whole answer, certainly not a satisfying answer, but a strong indication of the role that modeling can have in guiding experiments. And obviously there's a lot more to be done, and we would like at some point to have a good model of the ring. We would like when we get a structure, which we will have, I hope, fairly soon, of the ring interfaced with the snare complex, possibly with complexin. There will be a highly constrained situation that will perhaps allow us to model the energies and the forces and their distributions in a reasonably well-defined system. And actually, Fred Paulsay and I, we have a grant actually at École Normale Supérieure to support a postdoctoral fellow that would like to work on that, and if there is such a person, we would really like to know about it. So, okay, I did have this, so this just, I don't want to take the time to show how the models work, but just to say that, Brunner has made a major advance here by solving the structure of one of these binding sites, and so we're beginning to get the kind of information that can begin to answer some of the remaining questions. So I think I'll conclude just by thanking the people that contributed so much to this recent work. In my lab, Shyam Krishnakumar is a research faculty member who contributes enormously through his leadership of this project, and he and I and the group work very closely together. Jing Wang, I mentioned, as the person who did the electron microscopy and discovered the ring. Oscar Bello found the mutations that affect ring formation. Natalia Maria Zanetti, her name is also Natalia, carried out the experiments that demonstrated the generality of the synaptotagmin ring as an important principle. Feng Li, together with Jing, discovered that the rings polymerize in solution as pure proteins. The experiments that I showed you at the very first. Alexander, together with Yongli Zhang, is carrying out and has carried out important optical tweezer experiments. The electron microscopy and EM crystallography we're doing in a very close collaboration with Charles Sindelar, who's a faculty member at Yale in biophysics. And who else should I mention? Fred Ponce, I've mentioned his name several times already. And I mentioned Bourdain, who's started the physiology experiments with the PC12 cells. With David Baddeley at Yale, who's an engineer physicist who is developing sort of next generation super resolution imaging, we're optimistic that we'll get the technology to the point that when you have oriented rings on a membrane surface, we'll be able to see a donut structure with a hole in it, maybe under a synaptic vesicle if it's there. So we definitely aim to try to visualize that structure in living cells. And with Kirill Valinsky at UCL, we're carrying out experiments with him and his colleagues to test very specific mutations that we derive from the structure, such as the ring-forming mutation, to see exactly what they do in a much more physiological setting to the readily releasable pool, spontaneous release, asynchronous transmission, and all of the usual kinds of factors. So thank you very much for your attention.
Thank you, Jim. So are there any questions? So maybe we have now some time for a question session.
Do you think it's possible that the synaptotagmin rings could be disrupted by mechanical stretch of the membrane? Do you think that might be related to why hyperosmotic solutions can cause release of the...
Yeah, maybe that's true. I hadn't made that connection. It's a good idea. I mean, of course, they could be, and maybe they are. It's a really interesting point. I look forward to visualizing them, and then we can see. But it might be actually that... I mean, look, what we do find is that, preliminarily, is that it appears that the synaptotagmins are in clusters. That's been found before by Reinhard Jahn and colleagues. But it seems those clusters are busted up by the mutation that I'm referring to. So it might actually be that we could image and do that osmotic experiment even without, the highest level of resolution. Interesting. Thank you for that. It's a good suggestion.
So what would cause spontaneous release, do you think? Do you think it's like calcium ions in the cell, or do you think it's fluctuations of snares, fluctuations of other things?
Yeah, I with the disclaimer that I'm not an expert on this, and I asked that question just yesterday to Kirill Valinsky at UCL. Apparently he and his lab and probably other labs have experiments that say a lot of it, if not most of it, is calcium. So calcium, either a calcium channel opening spontaneously, I believe he mentioned that that's the largest cause of it, or also possibly calcium coming out of the endoplasmic reticulum, which is another local store. So that's one source, but apparently not all of it is due to that. And I'm guessing, like most things in neurophysiology, there's some debate, and probably different in different systems and all that. But so some of it can apparently be calcium. Others, I assume, is stress. I mean, I assume that if you have any system that's tightly wound, it's going to have some spontaneous excursion, and I assume that would be the remaining. Yep. Good question.
That was a wonderful talk.
Thank you.
You had this cartoon of lines of vesicles approaching the membrane, and in what sense are the vesicles sort of preparing themselves as they get closer and closer?
In every sense.
Can you
in every sense.
that?
I mean, people, I don't work on this, but there's certainly a lot of data over many years where people track individual vesicles, and they track a vesicle, after fusion, it recycles by budding inwards, and at that point you can get one or another dye or a particle inside, so you can track it. And you can say, okay, what happens to that vesicle? How long does it take to end up at the front of the line? What proteins are necessary to get from here to there? There's a lot of work along those lines, and it's a very complex, kind of machinery. It's not just a single step, but it's very clear that, broadly speaking, and it's an assembly line.
So the snares are being prepared as they get closer and closer.
Well, I mean, I think what's happening is that the snare again, there's a limit to what we know, what we have to infer, but I think most people would agree that the snare complex is now fully assembled after the vesicle fuses. The NSF ATPase comes in, separates the subunits. The VAMP molecule is separately collected into vesicles, separate from the T-snare subunits. And those vesicles are therefore reloaded with unfolded VAMP. Okay? But there are a series of upstream interactions, even before synaptotagmin. Okay? There are proteins like synapsin. They have all kinds of synap-type names. They form kind of a matrix, and the surface of the proteins on the surface of the synaptic vesicle kind of generally adhere. So the vesicles that you saw in the cloud are more or less bound to that protein that Paul Greengard discovered. That's regulated by phosphorylation. Everything that can be regulated is, and it relates to learning and memory. I mean, it's very complex physiology. And then there's another class of proteins. There are so-called RAB GTPases, which are a family of GTPase switch proteins that are important generally in vesicle trafficking. This vesicle is not deprived of one. It has its own. And that one binds to a tether protein, which is concentrated to a large degree at the active zone. That's called the ring protein. Okay? And again, Tom Sudhoff, I think principally among others, has established the role of this. I mean, it's a big field. A lot of people have contributed, but his contribution is really outsized. And so this vesicle, the rat, is initially kind of captured there by tethering. Still, the synaptotagmin needs to bind. Still, the snares need to bind. And there's probably stuff that we don't, at least that I don't know about. So it's very highly orchestrated. And if you think about it, it's probably the most highly orchestrated. It's certainly the most highly orchestrated fusion process, membrane fusion process, in biology. And it's quite possibly the most highly orchestrated regulatory process in the body. So anything that can be regulated is regulated.
Do you have an idea how the binding energy between two B domains of the synaptotagmin compares to the zipping energy of a snare complex?
Yes, it's much weaker. The only insight that we have into that comes from the experiments that Fang did, where he's measuring the rings forming in oligomers in solution. Okay, and so he can do that as a function of concentration. And assuming, and so there's, we measure the efficiency of conversion to rings, oligomers, as a function of the monomer concentration. Okay, and if you assume that the binding constant is not affected by the length of the ring, by the length of the polymer, which is a standard assumption, and reasonable, then you get a binding constant of about 10 micromolar in that range, something like that. Okay, which is not huge, far from it, but the local concentration of these proteins, when you have 20 of them all coming off of a vesicle, all oriented and collected into a patch of PIP2, which is itself highly clustered. I mean, actually, in the plasma membrane on the active zone, the PIP2s, as Reinhard Jahn and others have found, is in the form of a nanodomain, perhaps 80 or 90 nanometers, if I remember correctly, in diameter. And there are estimates of what percent of the lipid there is probably, certainly most of it is PIP2. So, and the T-snare syntaxin actually helps self-assemble that PIP2 domain through positively charged residues in its membrane anchor. So this whole system is very finely tuned to create favorable circumstances that drive these reactions even with relatively low affinity. Does that make sense? So the local concentration of synaptotagmin is likely to be many millimolar. Another good question. We are setting a very high standard with these questions, so whoever's next has to worry. Okay.
I just don't know whether this is going to measure up at all.
I don't either.
There are at least some cells in which the location of the release site seems reasonably stable.
What type of cell?
Well, a hair cell, for example,
Okay.
a photoreceptor or something like that, where it seems that the release occurs within within a fairly narrow range of location
Okay.
on the presynaptic membrane. Is it possible that these rings can be stable as well? Do they have to form every time a vesicle docks?
Oh, I
Could
see
they
what
be
you mean.
preformed and stay formed?
Well, okay. So it's really interesting. I'm not sure exactly what you're getting at, but let me see if I got it. One part of that could be, could the ring form from the synaptic vesicle even before the vesicle forms?
Yeah.
That's one possibility. But if you're asking, could the ring be stable and not come apart during the process of exocytosis? I mean, anything is possible, but that would not be consistent with our mechanism.
Yeah, no, I guess I'm not. Is there something that could hold the proteins in a general location so that the rate at which they form the rings could be accelerated because there's a local concentration?
Oh, yeah. That's, let me see if I can, the risk of going backwards in the talk. I mean, that's kind of what this is all about. These are all, and actually what I'm not showing you here is if you went out some distance from the active zone, you wouldn't see any vesicles. These vesicles are all collected in this region, okay? And when a vesicle is endocytosed, it quickly joins the pile from the outside, and it tries to get in. And so when I was responding to the earlier question about these vesicles, they're the ones that seem to fuse, and then they're replaced sort of in an assembly. It's not quite a rigid assembly line, but kinetically, but roughly speaking, and it's an assembly line. Does that make sense?
It does seem to be fairly rigid in a photoreceptor. They come down the ribbon, and the release site seems to be reasonably
Stable, right? Yeah, because probably, so now you're coming to things that I personally don't know so much about, but my impression is there are other proteins, none of the above proteins, whose names I can't remember. They have fancy Drosophila names. Maybe somebody here works on them. I don't know. They're very fanciful names that are involved in forming active zones that are found also in our brains, and they are required to form these stable active zone regions. Okay, and so I don't think there's to my knowledge, there's no structural concept yet as to how they work, but there are definitely proteins that do that, and they presumably interface in one or another way with this class of proteins to make sure they stay in the vicinity. So definitely that happens, no doubt, and you're absolutely right about that. But I mean, it's a very good question because you're kind of pushing to the outer limits of what we know. So, you know, to my mind, that's kind of the next level up in organization, but the organization is definitely there.
If you think of kiss and run mechanisms, you say that the snare proteins bring the vesicle and the membrane very close together, and then the ring of synaptic timing disassembles. How can the molecular machinery overcome this and form again a vesicle of the membrane, or is there just a kiss and run?
So what? That's a good. I'm not going to have probably a satisfying answer, but just as background for other people, when a vesicle first fuses with a membrane, it opens a hole, so to speak. You know, it communicates with the other side, so it opens a hole. That's called the fusion pore. And that fusion pore can expand completely to fully incorporate the vesicle, and that leads to the net growth of the membrane. So that is actually the mechanism by which plasma membrane, for example, grows, by acquiring the surface area of vesicles that fuse with it. Now, in a nerve ending, you don't want the nerve ending to grow in general, unless it's forming, okay? But in a mature circuit, you don't want it to grow. And so what you do, so that there's two possibilities. One is that the vesicle fuses, opens the fusion pore, through which the neurotransmitter rapidly exits by diffusion. And that, in fact, is the case. It exits very rapidly, within a millisecond or so. And then what happens after that? Well, one possibility is the vesicle pinches off again right there. The fusion pore closes off, and now the vesicle is separated. It can pump another cohort of neurotransmitter and reload, and now be ready again. That's called kiss and run. then full fusion is what you need to grow, okay? So if a nerve cell, if the synaptic vesicle fuses completely, then there's net growth, and that vesicle then needs to be recycled by endocytosis and in order to balance surface area and, of course, to bring the molecules back. Now, there's a big fight among synaptic physiologists of whether it's all one way or all the other way, and it's like, and maybe, or something in between. Actually, the year that Tom and I and Randy Sheckman shared the Nobel Prize, I was telling people this at lunch, the economics prize was given to 3 people who had 3 completely contradictory opinions on the very same topic. So in this respect, neurophysiologists are probably no better than economists. Okay? I mean, there was one guy from Yale, he's probably right because he's from Yale, and he's a nice guy besides. And he says that markets are irrational and driven by sentiment, and therefore there are bubbles. Then there's this guy from Chicago who's very rational, and maybe he comes from living in the Midwest, and he says actually everything is rational and there are no bubbles. And then there's the third guy who says, well, maybe they're both right. So anyway, they gave it to the topic. So I don't know who's right, okay, about that. But as I read the literature, it's probably a mix of both, okay? So then we're now down to the question, if you have, if it pinches off this way, how does that work, okay? Now my colleague at Yale, independent colleague, Erdem Karadikin, has measured with pure snare systems fusion pores. To my knowledge, it's one of the first, if not the first measurements. And what he sees is that pure snare proteins by themselves flicker. And they produce flickering fusion pores with considerable frequency. So, in other words, he's seeing kiss and run. So there's no question that kiss and run is an intrinsic property of snare proteins. So if there is no kiss and run in vivo in the synapse, there has to be some other protein that prevents it. Fred Paulsay and I have studied the fusion of liposomes, we've published this bit, with nanodiscs. And the nanodiscs are little chunks of lipid bilayer that are stable. They can be, 5, 6, up to 10, 15 nanometers in diameter, controllable. And so we put typically the T snare, or the V snare, doesn't matter which, into the disc, and then we let it fuse with a vesicle. Now, if you fuse with a disc, obviously the content goes through a pore, and when the pore first opened, you detect it in the medium, and that's what we published. You could do that. And what we found is that actually a single snare complex is enough. If you have a single snare complex in the disc, single snare in the disc, that's enough actually to open a pore, but then the pore closes off. It's kiss and run. Now the beauty of that experiment is that pore can't expand because it's in a nanodisc that's confined by protein and has a limited amount of lipid. Okay? And what we do find is that, as we haven't published this yet, when you get much bigger discs, we learned how to make bigger discs, now you get full fusion as the major product. But when you have a small disc, the fusion pore forms and closes off. So it is clearly a mechanism by which the snare proteins by themselves can lead to both full fusion, which of course, remember, they're used in cell growth. They're used in growth and division to make plasma membrane, okay? But they're also capable of flickering, okay? I hope that was probably a longer and more thorough answer but a very interesting subject.
So before closing the question session, I would like to ask you one question. You mentioned quickly during your talk that the calcium binding site of the synaptotagmin might be hidden between the membrane and the pocket. So would that...
Hidden in the interface between B and B.
So would that impede, how would that influence the calcium binding?
What I said, right, if you take what I said literally, and I do worry about this, then if it's truly buried in a protein-protein interaction, let's say that aspartic acid was forming a salt bridge to another residue on a separate domain of B. If that was really the case, then I think that would be a serious impediment to calcium binding. But we don't have the structural resolution to say that. We can only say what's in the region. So I have no reason specifically to believe that this region is not porous and open enough for calcium to come in and bind. But I do know that if the calcium binds there, in order to for the ring, in order to insert into the lipid bilayer, that subunit would have to rotate 90 degrees. And I assume it's breathing all the time, and I assume it's captured. It probably breathes and breathes and breathes, and at any one time spends most of its time in the ring, but occasionally one leaves and makes an excursion. And it may be actually during those excursions that it gets captured. I mean, that's another mechanism. I don't know. There's a lot to learn here. There's a lot of physical biochemistry to do.
Okay, thank you very much, Jim. We are very happy to have this wonderful talk, and now we are going to have a small reception, wine and cheese. So before that, let's thank Jim again. Thank you.